the chapter Excretory system from class 10. Last time we had covered with the parts of kidney longitudinal section, and today we would be doing in detail about the structure of kidney tubule. Okay, as I told you, this part is the longitudinal section of the kidney, and these parts were discussed last time. Okay, if I zoom in this part, you will see millions of small cells which are the structural and functional unit of kidney, which is called nephron, and one cell looks like this. So this is the structure of one nephron, right? And now today we would be discussing about its part. So let us start. You can see this structure is very complicated diagram. You can understand from the diagram only. And now what happens inside this structure is divided in basically this part, this shape, cup shape thing is Bowman capsule, right? I'm just going to tell you the parts first and then we'll come to the function. This is Bowman capsule. This knot like structure is glomerulus, which is also called capillary network. Along with that, you can see the arteries are coming into this glomerulus and then it is leaving. So the one which comes inside, that is efferent. Efferent means to get in, and the one which leaves this is efferent arteriole. Okay, how you can remember? E means exit, so exit one is efferent and the coming one is efferent. Okay, just see the difference in the width. So you can see here efferent one is quite wider compared to that of efferent. From the size you can easily make out. After that, now what happens? This Bowman capsule, you can see the loops are here. These loops are called PCT, proximal convoluted tubule, or first convoluted tubule, and then it becomes a long pin like structure which is called loop of Henle, and then again the same kind of structure similar to this PCT. This is called DCT. DCT is distal convoluted tubule, and then ultimately we have collecting duct. Now here one thing you have to remember, if you see the color change, the color of the capillaries changes from red to blue. It means that during the passage of filtration, the oxygenated blood becomes deoxygenated, because we already know that this red is for the oxygenated blood and blue one is for the deoxygenated blood. Now let us discuss these parts in detail. Okay, now starting from here, blood basically it comes from here and goes into this knot like structure. Now because this knot like structure is going to take up all the metabolic waste, along with that some important chemicals, for example glucose is there, and along with that we do have proteins and water, salt. These are being taken up by this part. I mean, this knot like structure is going to act as a suction pump, and it is going to take up all the important and unimportant things from this particular blood. Now you might be thinking, this is oxygenated blood, then why this we I'm talking about this thing, that is metabolic waste. Remember, metabolic waste blood and the other blood, which is your oxygenated and deoxygenated, don't confuse them. When I say oxygenated, I'm talking in terms of oxygen, means this particular blood has got more oxygen, or deoxygenated means it has got less oxygen. But when I'm saying that this particular blood has got waste doesn't mean that the blood is oxygenated or deoxygenated. We are not talking in terms of air, but we are talking in terms of the solid waste that this particular blood is carrying. Although the blood is oxygenated, but it is carrying metabolic waste. So if you have done with the circulatory system chapter, what happens? Heart, after pumping everything through aorta, it sends oxygenated blood to all parts of the body, and now this particular has actually come from the aorta, this particular vessel. Okay, now after that what will happen? I told you from this part everything that is important and unimportant things. would be taken up, right? We want the unimportant things to be thrown out of our body. What are those unimportant things? Nitrogenous waste. Now what are nitrogenous waste? I told you that was urea and uric acid. Okay, now when this would be done, you can see this width changes. And yes, one more thing, the blood from here comes under very high pressure, right? Goes in, and then again here when the blood enters this part, this particular thing has got thicker, means this particular blood will be thicker. Why? Because I told you water and some more things would be taken out. So here the blood would be thicker, so its consistency is not proper in this case. Right now what will happen, this unimportant and important things will going to travel here. You can see this arrow, you can see the arrow, right? Just I've zoomed in so that we can see. You can see the arrow going here, then here, and then ultimately coming somewhere here. Right now what is this arrow? Arrow is basically the things that are being taken up from the blood. I told you important and unimportant things. Okay, now what will happen? I told you your kidney has taken something which is not useful. Yes, that has to be thrown out, right? But along with that, I told you important things. Now body cannot throw important things out of your body. What are the important thing? I've already mentioned, it was glucose, protein, water, salt. Some things have to be retained in this particular blood, right? So what will happen, as this particular thing will travel, this things, what will happen, this is going to take, or you can say this thing is going to give something important back to these capillaries. That is why you see here these are capillaries. From normal arteries and veins, the exchange of material or gases is not possible, right? From arteries and veins. That is why here we have network of capillaries, right? And this particular network of capillaries is called vasa recta, right? It is called vasa recta. And in your book, if you see, it is written here secondary network, capillary network. It is written that in your book, right? So accordingly remember both the names. Okay, now yeah, so where I was talking about this thing, secondary one, it's written here very clearly. Okay, now what will happen, this particular part here, this convoluted means ubad khabad kind of structure, that is said to be PCT, right? That is proximal convoluted tubule. Full form could be asked in the exam, so don't leave any kind of full form in bio. Okay, now after that, this is also called first convoluted tubule. First, there is a reason why the word first and second has come, because similar kind of structure is seen here also, right? So this is first, that is proximal means close to this Bowman capsule, right? And this one, because it is away from this thing, that is why the word is second convoluted tubule, that is distal convoluted tubule is given to this particular structure. Okay, now after this PCT, we see a big hairpin kind of structure. This particular hairpin kind of structure is called loop of Henle. That is called loop. Here it is written loop of Henle, right? And also then after that, this loop of Henle goes to, this part is called, I told you, second proximal, second convoluted tubule or distal convoluted tubule, which ultimately becomes, ultimately turns to collecting duct. Not turns into, I mean it is connected to this part. Now here what all, first of all, structure should be clear to you in this particular part, right? I have not talked about ultrafiltration and filtration. I've just given you a overview, because that I would be doing it in the next class. Abhi, just remember the kind of structure. And yes, one more thing, remember the sequence. And it is not necessary that board is going to put sequence in, from starting from this, and then they'll ask you this. They might start from, say, first could be collecting duct, and maybe then they ask you write the sequence. So you might think, array, collecting ducts, okay, then I'll write Bowman capsule, then I'll write PCT, loop of Henle, DCT. Not at all. Remember, when they are asking collecting duct, so you will be going in reverse order. Reverse means first of all DCT will come, then loop of Henle, then PCT, then after that would be this part. So make sure, don't just look at the sequence part thinking that yes, sequence, I know the correct order. Remember in what order they are asking you, whether it is going forward or it's going backward, because many times Many students, they know the answer, but yes, they do mistake by thinking that the order would be from the beginning only. So don't do that in exam, right? Okay, now, so what all structures you have to remember? This part, right? And the capillaries, then we have PCT, then loop of Henle, DCT, and this. Okay, one more thing I would like to tell you here, that Bowman capsule means this part, and PCT means till this part, till this part. This part lies in cortex. Now you must be knowing what is cortex. Cortex, I told you, was the outer region of the kidney, right, which is a darker region, right? So this part actually lies in this. After that, this part, loop of Henle, right, and the collecting duct, this part, this lies in the medulla. Medulla is the inner portion of the kidney, lighter in color. And yes, because these are just like, you know, lines kind of structure, they have got loop of Henle also and collecting duct. That is why it gives stripped appearance to the medulla region, that is kidney region. So remember, this question could be asked, that why medulla has got stripped appearance. So your answer would be because of the presence of loop of Henle and collecting duct. It gives it a appearance as if it is a line kind of. Stripped means like this, right? Okay, so we'll be covering the next portion, that is the function of kidney, ultrafiltration and reabsorption, in the next video. And do revise the question. I would be putting up some questions for practice from this part.